Hello, everyone, and welcome back to Microbiology. In this lecture, we will be covering part 2 of Chapter 8, Microbial Metabolism. The lecture objectives for this lecture are to remember key components of metabolism, reaction coordinates, and metabolic classifications, understand the role of energy, ATP, and electron carriers play in metabolism, apply these to carbohydrate metabolism, including glycolysis, fermentation, and cellular respiration, analyze the flow of carbon, energy, and electrons in these processes, evaluate fermentation tests, and categorize microbes based on the results, and apply the knowledge of the cellular electron transport chain to the light-dependent phase of photosynthesis. Let's review some key concepts before we continue further. As previously discussed, metabolism refers to the chemical reactions building up and breaking down nutrients within cells. Catabolism is a process that breaks down complex molecules into simpler ones and releases energy. Anabolism builds up complex molecules from simpler ones and usually requires energy consumption. These two processes form a cyclic pathway where energy is continuously cycled between catabolism and anabolism. The process of catabolism involves the oxidation of molecules to extract their chemical energy. Specifically, in this case, glucose is oxidized to CO2 and water molecules. This process will be discussed in more detail later, but oxidation in general means the taking of electrons from. And oxygen is very good at taking electrons. So we often see oxygen involved in oxidation, and it's where the name oxidation, where the taking of electrons comes from. The energy released during this process is harnessed to produce ATP. In contrast, anabolism uses the ATP generated during catabolism to synthesize more complex molecules. In this instance, amino acids serve as the building blocks for the synthesis of proteins, as you may recall from previous coursework. Activation energy, represented as E sub A, is necessary for certain chemical reactions to occur, acting as a spark to initiate the process. This metaphorically means that a little bit of energy is required to kickstart the reaction. An excellent illustration of this is the burning of wood, which contains carbon-hydrogen bonds that, when burned, convert into carbon-oxygen bonds and carbon dioxide. Again, this is a form of oxidation on a much quicker scale than what we see in cells. And uh, carbon dioxide stores less energy. It's in a lower energy form. As a result, a considerable amount of energy is lost from the chemical bonds. In this example, it is lost as heat. And in cells, an attempt is made to capture the energy as ATP in general. Although this reaction results in more stable byproducts, the carbon-hydrogen bonds with lower energy, it necessitates some heat or activation energy to get the process started. To aid in this understanding of the concept, we can plot a reaction coordinate, seen on the left here. It tracks the reaction's progress in relation to the energy present in the chemicals, including the reactants here in the beginning, the transition state in the middle, and the products. The reaction coordinate is an excellent tool for illustrating this process, with free energy represented on the y-axis. Upon examining the coordinate, the reactants are located to the right and the products are located to the left at a lower and more stable energy level. In the case of the campfire, wood is the reactant and carbon dioxide is the product. The line in the coordinate demonstrates the amount of free energy in the system. We can see here, this line here, called G, or Gibbs free energy. And the change in free energy is represented as delta G right here, with the Greek triangle sign representing delta, which means the change of. The transition state indicates that some free energy must be added to the system in order to initiate the reaction. However, overall, delta G is negative. So here's the reactants, and there's an energy release. So it's being removed or subtracted. So the delta G is negative, and this indicates greater stability of the products versus the reactants, and an energy release. The chemicals must, however, first go uphill to get their required input energy. This is the activation energy. For instance, in the campfire example, we add heat to initiate the process, and eventually the reaction's own heat, released from the conversion of reactants to products, acts as the activation energy to convert more carbon-hydrogen bonds to more CO2 bonds. Activation energy can hinder reactions from occurring efficiently within the cell, causing them to take a long time to happen on their own. Life has evolved enzymes to assist in this process and reduce the activation energy required for reactions to occur in a timely manner. Enzymes work by stabilizing the intermediate step of the reaction coordinate, reducing the activation energy required for the reaction to occur. This allows the same change in free energy, or delta G, but a faster reaction, ensuring that the cell can execute reactions selectively and regulate energy processing by using enzymes to pick and choose which reactions happen at what rate. By stabilizing the transition state, enzymes enable reactions to require less activation energy, which is the sole reason why enzymes speed up reactions. ATP is a molecule that can store free energy for later use in the cell. It's a usable form of energy. Upon hydrolysis, it becomes a more stable form called ADP and releases some of its stored energy, similar to how wood converted into CO2 in our prior example. However, instead of dissipating the energy into the environment as heat alone, the released energy is channeled towards specific reactions in the cell. For instance, the energy can be used to drive anabolism, the process of building complex molecules from simpler ones. In this way, ATP serves as a vital energy source for the cell, facilitating various metabolic processes necessary for cell survival and growth. Recall that we have two components we need to keep track of when classifying the metabolisms of organisms. Together, our metabolic classification identifies the way in which an organism obtains its energy and carbon sources. For instance, a photoautotrope harnesses energy from the sun and obtains its carbon from inorganic sources, whereas a chemoautotrope obtains its energy from carbon from and from inorganic sources. In contrast, animals are restricted to chemoheterotrophy and do not possess the same diversity of metabolic pathways as other organisms. While animals are structurally fascinating due to their multicellularity, and behavior is incredibly interesting as well, their metabolic chassis is limited in terms of energy and carbon source acquisition. The classifications mentioned above are exceedingly wide-ranging, considering the diverse means by which living organisms derive their livelihoods, from the hydrothermal vents on the ocean floor to the microbes inhabiting our digestive tracts. We can explore these topics in great detail with meticulous attention. We will first turn our attention to carbohydrate metabolism. In the world of microbes, obtaining energy is essential for survival. Microbes mainly acquire their energy by breaking down carbohydrates, such as glucose, through either cellular respiration or fermentation pathways. These pathways both involve the oxidation of glucose to generate ATP, which is the primary energy source for most cells. Essentially, the two pathways help microbes to convert glucose into usable energy that can be used for various cellular processes. That usable energy being ATP. This lecture may be disorientating, but focusing on some key questions should assist you in navigating the metabolic processes. To make it easier, we can focus on some key questions that will help us understand the underlying mechanisms. And these are the questions I want you to think about when we go through each slide as we cover each of the components of metabolism, of carbohydrate metabolism. These questions include determining where the carbon is going, whether molecules are being built or degraded, the number of carbons involved, and where the energy is being directed. We can also determine whether ATP or ADP is being produced, if energy is being spent to create complex molecules, or if energy is being harvested by breaking down molecules. Additionally, we can examine where the electrons are going, such as whether they are being transferred to oxygen through oxidation or carried by electron carriers. By considering these questions, we can gain a better understanding of the metabolic processes at play. We will begin our coverage of carbohydrate metabolism with glycolysis. The process of glycolysis involves um, the breakdown of glucose into pyruvate. And if you are keeping track of your key questions, this would be an example of catabolism. Pyruvate can then enter one of the two pathways, cellular respiration or fermentation. The choice between cellular respiration and fermentation is determined by the oxygen requirements of the organism and the availability of oxygen, as cellular respiration requires oxygen while fermentation does not. In cellular respiration, pyruvate is oxidized and transported into the mitochondria. There, it undergoes the Krebs cycle, which harvests the electrons. The final stage is the electron transport chain that produces a significant amount of ATP. In order to continue with this conversation, we need to understand a little bit more about FADH2 and NADH. These are molecules that carry electrons produced during the breakdown of glucose and other molecules in the Krebs cycle. These electrons are used to generate ATP in the electron transport chain, where they are used to pump protons across the mitochondrial inner membrane to create an electrochemical gradient that drives ATP production. And we'll see that in the coming slides. These electron carriers have different names depending on if they are carrying or not carrying these electrons. NAD plus is not carrying the electrons, and so it can act as an electron acceptor where it can take on these electrons and become NADH. So if it acts as an electron acceptor during the oxidation of glucose, then it then becomes NADH, which contains the extra electrons. NADH can then act as an electron donor for the electron transport chain, which is the last step in cellular respiration. We are mostly focusing on the metabolic pathway of cellular respiration, but NAD+ is an essential coenzyme for many enzymes involved in catabolic reactions, while NADH is important as an electron carrier that delivers high energy electrons to the electron transport chain for ATP production. Together, these molecules allow cells to extract the maximum amount of energy from glucose and other molecules to support essential cellular processes. In a similar fashion, we have FAD and its electron carrying counterpart, FADH2. This image provides a high-level overview of cellular respiration. Now don't worry, we will explore these steps one by one, but sometimes it's nice to see the final destination so we understand the road we take to get there. Each and every one of these steps is catalyzed by a single enzyme that reduces the activation energy of that reaction. At the top left, we have glycolysis. This occurs in the cytoplasm. Glycolysis breaks down glucose to give off ATP and NADH, finally leading to pyruvate at the bottom here. Following glycolysis, pyruvate is oxidized, which generates acetyl-CoA, and that acetyl-CoA then enters the mitochondria, shown at the bottom here. Acetyl-CoA is now in the mitochondrial matrix. Actually, pyruvate oxidation also occurs there as well, and the Krebs cycle takes place within the mitochondrial matrix, which is kind of like synonymous with the cytoplasm of the mitochondria. It's this inner fluid. Okay. The Krebs cycle then metabolizes acetyl-CoA through a series of steps to harvest more electrons, generating NADH and FADH2. Finally, these electron carriers make their way to the inner membrane of the mitochondria to synthesize ATP in the electron transport chain. Glycolysis is the metabolic pathway that converts one molecule of glucose into two molecules of pyruvic acid. You may see pyruvic acid also called pyruvate, but for all intents and purposes are describing the same thing. Along the way, two NADH and two ATP are generated. And remember, we're keeping track of both of these molecules as well. So here we have a catabolic reaction going from a more complex molecule to two simpler molecules. We have captured energy from this reaction in the form of ATP, and additionally we have transferred some electrons to NADH. Overall, the net result of glycolysis is the conversion of one glucose molecule into two molecules of pyruvate, two molecules of ATP, and two molecules of NADH. Finally, let's analyze where the carbon is going. In the overall metabolic pathway, we can see that glucose is a 6-carbon molecule, and it is split in half into two 3-carbon pyruvic acids. In eukaryotes, pyruvate has to be imported from the cytoplasm of the cell into the mitochondrial matrix for further processing. Once there, it is further oxidized to create acetyl-CoA. This is an oxidation reaction that releases one of the carbons of pyruvic acid as CO2, and where we again transfer electrons to NADH. The overall result is for every pyruvic acid molecule we generate, we get one NADH, one CO2, and one acetyl-CoA. Since one glucose molecule is split into two pyruvic acids, for every one glucose molecule, pyruvate oxidation will generate two NADH, two CO2, and two acetyl-CoA molecules. Keeping tally on our carbon flux, let's recall that pyruvic acid has 3 carbons in it. So we can see that we have 6 carbons altogether in our two pyruvic acid molecules. We lose 2 of these 6 carbons as CO2. Acetyl-CoA has 2 carbons in it, and we are therefore left with 4 carbons in our two acetyl-CoA molecules. Checkpoint 1. How many pyruvic acids are produced from glycolysis? And then, how many acetyl-CoAs are produced in the following pyruvate oxidations? The Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid cycle, is called a cycle because it is a series of enzymatic reactions that occur in a cyclic manner. The cycle starts with the product of pyruvic acid oxidation, acetyl-CoA, which enters the cycle by reacting with oxaloacetic acid, thereby generating citric acid. It undergoes a series of chemical reactions, eventually regenerating the starting molecule oxaloacetate. It's not necessary to memorize all of the intermediate products, but keep track of the key questions we outlined in the beginning of the lecture. Our starting materials are 2-acetyl-CoAs, which has a total of 4 carbons. And in one cycle, we lose 4 carbons. In the 2 cycles altogether, we lose the 4 carbons as carbon dioxide. We have now accounted for all of the carbon in this carbohydrate pathway, and all of it escapes as CO2. For each glucose molecule, we lose 2 carbons of CO2 in pyruvate oxidation, and the other 4 carbons is CO2 in the Krebs cycle. One important thing to note in the Krebs cycle is the incredible number of NADH and FADH2 that are generated. Processing both acetyl-CoAs has left us with a whopping 6 NADH and 2 FADH2. This is where the majority of electrons have been harvested. Lastly, there is a new molecule we haven't discussed, and that is GTP. GTP is just another energy currency in the cell. Just like ATP, it's just not used as frequently. The Krebs cycle is one of the examples where it is actually produced. And it's about as equivalent as an ATP for cellular functions. And we get 1 ATP for each acetyl-CoA that goes through the Krebs cycle, amounting to a total of 2. So things to memorize from the Krebs cycle include, for each glucose molecule, the number of acetyl-CoAs that enters the Krebs cycle, the number of CO2s that leave it, as well as the total NADH, FADH2, and GTP, in other words, ATP equivalents that are synthesized. Let's take a step back and put together all of the electrons we have harvested along the way per each glucose. We received 2 NADH in glycolysis, 2 NADH in pyruvate oxidation, 6 NADH along with 2 FADH2 in the Krebs cycle. That's 10 NADH and 2 FADH2 that are primed and ready to donate their electrons to the electron transport chain to produce ATP. Here we have the final step in cellular respiration, called the electron transport chain. As the name implies, this is a chemical process of transporting electrons. It occurs along the inner membrane of the mitochondria in eukaryotes, or simply on the plasma membrane of prokaryotes, where you can see the phospholipids in the phospholipid bilayer of that membrane. You can see in the red boxes NADH and FADH2 highlighted. They each donate their electrons and hydrogen, also called a proton, to the electron transport chain, either through these complexes such as NADH dehydrogenase or through membrane-bound electron carriers. These carriers are just membrane-bound and they move it from one complex to the other. Quinone is listed here, another one is ubiquinone, but you do not have to worry about memorizing those, just the process. So you won't be required to memorize these proteins. But you can see it creates a chain of electrons. These electrons get passed through this chain here. And these electrons are used in order to pump protons across the membrane. See, these protons get moved from NADH through the membrane, same with FADH through the membrane. And they eventually land in what's called the intermembrane space, which is the space between the inner membrane and outer membrane of the mitochondria. Take a moment and locate where oxygen is on this electron transport chain. And let's see if we can make some sense out of what the role of oxygen is in this system. So if we look up here, here's oxygen. Something we should get used to understanding about oxygen is that it loves electrons and therefore will readily react with them in its presence. In fact, the process of oxidation gets its name from oxygen, which is the process of taking electrons. We can see here at the protein cytochrome oxidase complex that an oxygen molecule reacts with electrons and protons to create water. The name for a molecule that accepts electrons at the end of a metabolic pathway is called a terminal electron acceptor. So oxygen is the terminal electron acceptor, and that's its major role in cellular respiration. The protons are used by ATP to synthesize, to generate, or sorry, it's used by ATP synthase to generate ATP. One glucose molecule will use all of those NADHs and FADH2s to generate 34 ATP from the electron transport chain, or ETC. So why is glycolysis and cellular respiration so important for us to know? Well, imagine what you are doing right this moment. You are breathing in oxygen, and you are also breathing out CO2. This is the process of cellular respiration. You're breathing out CO2, which is actually the byproduct of pyruvate oxidation in the Krebs cycle. Additionally, the oxygen you breathe reacts with the electrons in the electron transport chain and two protons creating water, and thereby having a final resting place for the electrons coming from cellular respiration. Therefore, you can see that this knowledge can be applied to various fields of study, including biochemistry, physiology, nutrition, and medicine. Moreover, by building on this understanding, we can determine how factors affect glycolysis and cellular respiration, and we can make informed decisions about our lifestyle choices and take steps to improve our health and prevent disease. Here we can see where the electron transport chain occurs in prokaryotes and mitochondria. In prokaryotes, the protons are pumped from the cytoplasm to the outside of the plasma membrane. For gram-negative bacteria, this would be in the periplasm, and for gram-positive bacteria, this would be between the plasma membrane and the peptidoglycan. For mitochondria, the protons are pumped across the inner membrane from the mitochondrial matrix to the intermembrane space. Checkpoint 2. In which stage of cellular respiration are the majority of electrons harvested for later use? Hint: ATP synthesis uses those harvested electrons. So, what is the purpose of all of this? Surely it isn't just to make water from electrons, protons, and oxygen. No, ultimately, this is to produce energy. As protons build up on one side of the membrane, the intermembrane space has a higher concentration of protons than a mitochondrial matrix. Ultimately, the proton concentration wants to be balanced on each side of the membrane. To do so, they must pass through a membrane-bound enzyme called ATP synthase. Think of it like a hydroelectric dam, only instead of using water, we're using protons. And as protons pass through ATP synthase, it spins the turbine and converts ADP to ATP synthase. You do not need to remember the other membrane proteins in the ETCs, but you will need to know ATP synthase due to its key role in the process of ATP generation. Now let's apply our knowledge to understand a real example. I want you to imagine what would happen if we had a depletion of oxygen in the absence of oxygen. In the absence of oxygen, the accumulation of electrons would cause the electron transport chain to be congestion, correct? This would lead to a shutdown. This would prevent the production of the majority of our energy in the form of 34 ATP. Instead, we would have only Oh, only about 2? Yes. 2 ATP from glycolysis and 2 GTP from the Krebs cycle. However, even with this, we might run into an issue as well. But this would be insufficient to meet the energy demands of our cells on a regular basis. This would place our cells in a precarious situation, unable to generate enough energy to support their vital functions. Let's now analyze the flow of NAD+ and NADH across the whole set of reactions in glycolysis and cellular respiration. Through glycolysis, NAD+ takes electrons to form NADH. This NADH is put to work to synthesize ATP in the electron transport chain, and this regenerates NAD+ so that the cycle can start anew. However, we need oxygen as the terminal electron acceptor. Now, not all life on Earth lives in oxygen-rich environments. In fact, places like our digestive tracts, deeper layers and in the soil, or lower in the water column, there may be an absence of readily available oxygen. What now? If there's not oxygen, we cannot get rid of the NADH and its electrons. And then we lack the NAD plus to begin the process again with glycolysis. We've discussed how disastrous this can be for us in the electron transport chain. But have organisms evolved any alternatives? The answer is yes, and one of those alternatives is called fermentation. So imagine this is all gone, and we can no longer, without cellular respiration, use those 36 ATP. But at least organisms can live in regions inhospitable to others. They ultimately are limited to just 2 ATP per glucose molecule through fermentation. But what is fermentation? You may have heard of fermentation colloquially in some of the processes we use it in. But fermentation is an alternative metabolic pathway under anaerobic conditions or conditions without oxygen, which instead of using oxygen as the terminal electron acceptor, uses organic molecules as a terminal electron acceptor. And this regenerates NAD+ so that glycolysis can continue to run and produce ATP. Although fermentation generally generates significantly fewer ATP molecules compared to cellular respiration, it operates at a rapid pace and can occur in the absence of oxygen. In this example, the 6-carbon glucose is converted to 2 3-carbon pyruvic acids that lose 1 carbon each as carbon dioxide, and the final product is 2 ethanol molecules. Ultimately, no carbon has been lost. We started with 6 carbons and end up with 6 carbons. How can an understanding of fermentation benefit us, though? Well, understanding which microbes can grow in the absence of oxygen can help in their identification, which can be useful in many fields, including microbiology, including medical uses when identifying the presence and type of a pathogen. Fermentation is also utilized in industrial processes, such as the production of alcoholic beverages and the manufacturing of bread and dairy products. Finally, understanding the chemical reactions and pathways involved in fermentation can provide insights into metabolic diseases, such as lactic acidosis, which occurs when the body cannot produce enough ATP via cellular respiration and switches to fermentation as an alternative energy source. Additionally, knowing the relative ratios of glucose to CO2 production can help prevent large capital losses due to poor foresight within these industries, as we are about to see. So this video right here is a video of a wine fermentation vessel. And if they had done the math, they probably should have realized that the amount of glucose available in that unfermented batch would have produced so much CO2 that this was going to basically destroy this batch of wine. Remember, we're getting 2 CO2 molecules per each glucose molecule. And that is wine under a lot of CO2 pressure. Okay, we'll leave it at that. As discussed, ethanol fermentation converts glucose to 2 ethanol molecules. Ethanol is only 2 carbons long, and the remaining 2 carbons are lost as CO2. Another type of fermentation we should be familiar with is lactic acid fermentation, where the end product is lactic acid. Lactic acid is 3 carbons long. Therefore, when glucose, a 6 carbon molecule, is converted to 2 lactic acids, we would not predict any CO2. No CO2 is made as a byproduct, as all the carbon is still kept in the organic molecule's lactic acid. Here is a summary for studying purposes of the different steps and processes of carbohydrate metabolism discussed today, and the important numbers and locations of them, of where they occur, to be mindful of. Checkpoint 3. List the total combined ATP and GTP created from aerobic respiration and how much from lactic acid fermentation. Here is a list of the different types of fermentation, the chemical byproducts from their organic terminal electron acceptors, and the commercial products that utilize these metabolic pathways. We've discussed lactic acid fermentation, alcoholic fermentation. There is also something called mixed acid fermentation, which leads to the production, namely, of acetic acid, which is vinegar. There's also propionic fermentation, which produces propionic acid, and it's used in the production of Swiss cheese. We have butane-diol fermentation, which produces butane-diol, gives Chardonnay, imparts a Chardonnay flavor to wine, and butyric acid or butyric fermentation, which produces butyric acid, which is what gives things a buttery flavor. Lactic acid is a key end product of the anaerobic metabolism called fermentation, um, called lactic acid fermentation, that can be found in various organisms. Despite being considered a waste product, lactic acid has many commercial applications, particularly in the food industry. It is a crucial component in the production of foods such as yogurt, pickles, and sauerkraut. These fermented foods are created by the conversion of sugars in the food substrate to lactic acid through the metabolic activity of lactic acid bacteria. In addition to its use in food production, lactic acid is also an important component of the gut microbiome, where it is produced by certain bacteria during fermentation of dietary carbohydrates. This production of lactic acid is a vital process in maintaining gut health and preventing the overgrowth of harmful bacteria in the intestinal tract. Ethanol is another key end product of anaerobic metabolism that has important commercial and biological applications. It is a primary component in the production of alcoholic beverages such as beer, wine, and spirits, which are created by the fermentation of sugars by yeast. Additionally, ethanol has emerged as a promising alternative to traditional fossil fuels in the production of biofuels. Like lactic acid, ethanol is also an important component of the gut microbiome, where it is produced by certain bacteria during fermentation of dietary carbohydrates. This production of ethanol plays a vital role in maintaining gut health and preventing the overgrowth of harmful bacteria in the intestinal tract. However, it's important that there is a well balanced ecosystem in our gut microbiomes. Auto brewery syndrome is a pretty unique condition where an overgrowth of microbes that focus on ethanol fermentation inhabit your GI tract, and your gut becomes a personal brewery in a sense. This can make you feel drunk without even touching a drop of alcohol. So basically, you could be a walking, talking distillery. Sounds pretty cool, right? But hold on, before you start throwing parties, this condition can also leave you feeling hungover, tired, and people have been given a DUI because of it. Now let's examine how we can use tests to try and identify organisms. A fermentation test. A Durham tube is a small test tube inserted into a larger test tube. You can see it here, and it's inserted upside down. Um, it's inserted into a larger test tube that has a sugar containing broth to test for fermentation of different types of sugars. The smaller tube collects any gas produced by the fermentation process, such as carbon dioxide, and traps it inside. This gas production can be seen as a bubble inside the tube. A pH indicator is added to the broth to determine if fermentation has occurred. If fermentation occurs, the color of the pH indicator changes from red to yellow. This helps to confirm that the microorganisms in the broth are capable of fermenting the particular sugar present in the broth and producing acids or alcohol and gas, which can have important applications in microbiology, such as identifying bacterial species in a patient and assessing the quality of food and beverage products. Here's an example of fermentation test outcomes. In example A, we have no acid produced and no gas. It's a lighter color here, but that's just because it's not as the diameter is not as big as where there's the full liquid here. But there's the liquid is still there. There's no bubble. In this example, it's hard to tell, but there is a bubble here. It's a little bit lighter than this one. In example B, there's gas produced but no acid. So in example B, this could be potentially ethanol production, right? There's a gas produced, no acid produced. In example A, it's hard to tell. Maybe there was just no fermentation at all. In example C, there's no gas, but there is acid production. Okay, so this could be lactic acid fermentation, right? And then finally, in example D, there is gas and there is acid. So this must be maybe a mixed fermentation or something like that. Acid fermentation. Checkpoint 4. Which outcome would result from the culturing of a microbe from the Lactobacillus genus? And in case you can't tell which ones have gas or not gas, I wrote below so that they're clearly identified. So fermentation is being utilized by genetic engineering in order to produce a variety of different end products. For example, scientists are using yeast and different types of bacteria that can ferment sugar in order to produce products that have a high commodity value, such as the perfume that smells like rose. And Ginkgo Bioworks is one of these companies working on this problem here. Additionally, fermentation can be used in order to produce the proteins that are found in spiders' silk fibers. And these are just a couple of examples, but there are just a tremendous amount of examples out there. Regrettably, due to time constraints, we cannot delve into the fascinating and peculiar realm of photoheterotrophs or chemoautotrophs. However, if any of you want to read further in the textbook or pursue careers in industrial, ecological, environmental microbiology, you may have the opportunity to explore these concepts in greater depth. Thus far, our discussion has centered solely on chemoheterotrophs. Nonetheless, before concluding, we must briefly touch upon the topic of photoautotrophs. Instead of using energy already stored in chemical bonds like chemotrophs, phototrophs use photosynthesis to harvest energy using light. They include algae, some protozoa, and cyanobacteria. These organisms, if they are photoautotrophs, are able to convert sunlight energy and carbon dioxide into chemical energy in the form of food. During the process, light energy is absorbed by pigments such as chlorophyll, which are present in specialized organelles called chloroplasts. This energy is then used to convert carbon dioxide and water into glucose, a simple sugar that can be stored and used later as a source of energy. The ability of photoautotrophs to perform photosynthesis is critical to the survival of many ecosystems on Earth, as it is the primary means by which energy enters the food chain. It also plays an important role in regulating the amount of carbon dioxide and supplying the atmosphere with oxygen, which is a critical factor in maintaining a habitable environment for many organisms, including humans. Without photosynthesis, life as we know it would not be possible. During photosynthesis, 6 molecules of CO2 combine with 6 molecules of water by using light energy that is harvested by pigment molecules such as chlorophyll to produce 1 molecule of glucose and 6 molecules of oxygen. You will be required to know this chemical formula. You must be able to write out 6CO2, 6H2O to create C6H12O6 plus 6 O2 molecules. And this is the basic overall picture of photosynthesis. It's actually the exact reverse reaction of cellular respiration, if you think about it. Cellular respiration goes in the exact opposite direction. And so this is another big cyclical paradigm. Checkpoint 5. Write the complete chemical equation for photosynthesis. The process takes place in 2 main stages or phases: the light-dependent reaction and light-independent reactions, also known as the Calvin cycle. During the light-dependent reaction, ATP is synthesized, and during the Calvin cycle, organic molecules such as glucose are synthesized using CO2 and the ATP generated from phase 1. During the light-dependent reactions, light energy is absorbed by chlorophyll in the thylakoid membranes of the chloroplasts, which causes the splitting of water molecules into oxygen and hydrogen ions. Hydrogen ions are it's the same as calling them protons. In a way, this is the reverse effect of the electron transport chain of cellular respiration. As was done in the electron transport chain of cellular respiration, the protons or hydrogen ions are used to create a proton gradient across a membrane and then to generate ATP using ATP synthase as the electron transport chain of photosynthesis. The oxygen released as a byproduct during this process is released into the atmosphere. In the Calvin cycle, short-term chemical energy and electrons, which are your ATP and your NADPH, it's a little bit different. It's not NADH in photosynthesis, it's NADPH. If it helps you remember, just think the P is for photosynthesis, are converted into long-term energy storage molecules such as glucose or other materials for the photoautotroph to use later on. As the Krebs cycle occurs in the mitochondrial matrix, the Calvin cycle occurs in the stroma of the chloroplast. The stroma is just It's a liquidy, gooey substance found inside of the chloroplast, just like the mitochondrial matrix is a liquidy, gooey substance found inside of mitochondria, just like the cytoplasm is a liquidy goo found inside of the cell, such as a prokaryotic cell or a eukaryotic cell in general. Those are just terms for those that matrix of goo. In the okay, finally, let's discuss the Great Oxygenation Event, which occurred approximately 2.45 billion years ago and was a pivotal moment in Earth's history. It was during this time that cyanobacteria, the first organisms to carry out photosynthesis, began producing oxygen as a byproduct of photosynthesis. This influx of oxygen into the atmosphere over time, a short time in a geological scale, led to the formation of the ozone layer, which helped protect the planet from harmful ultraviolet radiation. However, the rise of oxygen also had a significant impact on life on Earth, causing the first ever mass extinction, as many organisms were unable to adapt to the new atmosphere. Before this event, essentially the whole planet was anaerobic, and organisms were incapable of metabolizing the toxic reactive oxygen species. Overall, the Great Oxygenation Event marked a major turning point in Earth's evolution, paving the way for the development of more complex forms that require the energetic efficiencies of cellular respiration. And we'll close with that. That concludes the second part of Chapter 8. Thank you all for making it through, and please join me for the next chapter.